In our final content unit, we're going to be discussing what happens throughout the process of embryonic development, fetal development, all the way up through death. But in this first standard, we're focusing on the events of fertilization and ways that couples or individuals can seek treatment if they're having issues with infertility. FETUS.1 reads, I can describe the events of fertilization, including assisted reproductive technologies. Okay, so fertilization. As humans grow, they develop and age, but there's a difference between growth and development. Growth is just an increase in size. It's usually due to cell division, so you get more and more cells, which appear as bigger and bigger in size. Development, on the other hand, is how those cells are changing. So what is that continuous process as an individual progresses through their lifetime? So growth is something that happens for the first couple decades of our lives, but development continues on for the entirety of our lives. We're going to use a couple terms to describe those periods. The prenatal period is everything from that initial fertilization of sperm and egg through birth, while the postnatal period is only going to highlight birth through death. And by only, I mean those roughly 80 years. Okay, fertilization, also known as conception. So what's happening here is we are uniting a secondary oocyte, so after those 2 rounds of oogenesis, and one sperm cell of the hundreds of millions in an ejaculatory emission. We mentioned in the last unit that this takes place in one of those fallopian tubes, and before that can occur, we have to release a secondary oocyte from the ovary, and it only lives for about 12 to 24 hours after ovulation, meaning that the window for a woman's fertility is very, very small. However, sperm can survive up to 6 days in the reproductive tract, and a woman can't accurately predict when those 12 to 24 hours after ovulation are, so accidental conception or non-purposeful conception is extremely common. What are the events? What are the steps? So first, we have multiple sperm that will reach the egg. Of the hundreds of million, it'll only be a few hundred that reach the egg itself. And it's not the first one there that will necessarily be the one that fertilizes. So they come in and they invade this layer called the corona radiata. It's a layer of protective cells that need to be dissolved using the enzymes in the acrosome of those sperms. So that's what's seen happening here. These tiny dots are the digestive enzymes that eat away at those follicular cells. After that, they have to get through the zona pellucida, which isn't a cellular layer, but it's a sugary protein, glycoprotein layer that is lining the oocyte as well. So here it's shown in blue. All the sperm at this point are going to work together to remove those layers, the corona radiata and the zona pellucida. They're going to use each other's collective enzymes, but only one will make it. So as I mentioned in step 3, only one sperm will penetrate the egg itself. So there's that cell membrane of the secondary oocyte, and this sperm cell is the one that will fertilize. When that happens, the zona pellucida, this blue region, changes molecular composition and hardens, effectively preventing any other sperm from entering the egg. So once the first one gets all the way through that zona pellucida, then that's it. Everything seals off because we can't have too many chromosomes inside that egg. Next, the sperm will sort of degrade, and its nucleus will unite with the nucleus of the oocyte. So we have 23 chromosome pairs. So each pair, one from mom, one from dad, will find each other. The nucleus that forms here is called a pronucleus, which means like initial, almost like prototype of the eventual nucleus of this individual, this brand new set of chromosomes. Once this happens and those two pronuclei come together and form a little ultimate zygote, we have our blueprint. So we can start to divide that cell and differentiate it into the eventual tissues of a mature human body. A zygote is the name we use for the combination cell that has all 46 chromosomes but is a singular cell. It's basically a fertilized egg. Okay, so what can we do if a couple or an individual is having trouble conceiving? Infertility is defined as not being able to get pregnant despite trying and having unprotected sex for at least a year. Estimates say that 1 in 5 couples experience infertility, and those numbers appear to be rising as couples wait longer and longer to have children. About 1% of US births result from one of these methods we'll discuss, these assisted reproductive technologies, or ARTs. Okay, so what do we do if we're having trouble delivering the semen? If that's the problem, maybe it's an erection issue or an ejaculation issue, or a woman wishes to have a child without the aid of a male partner, then in this case, we use donated sperm and intrauterine insemination. This is where the doctor uses a catheter to place donated sperm in a woman's reproductive tract. Again, like I said, we can use it if the male is infertile, in which case we need a different male's donor sperm, and it will not be this original male's child. Maybe if the male has a genetic disease, if he has a low sperm count, erection or ejaculation issues, or I said either a same-sex female couple or a single woman. Its success rate is about 20%, so it's not super effective. However, it is relatively cheap, and so rounds of this can happen before you accumulate a huge bill. We're talking $500 to $1,500, and I said that's cheap. What about IVF, in vitro fertilization? This is where we take the egg and the sperm and we unite them in a petri dish. So fertilization occurs outside of the woman's body, and then we place that embryo into the uterus artificially. We can use this if a woman's having issues with her fallopian tubes, like a blockage, or if a couple wants to select a specific embryo to implant. So if there's a family history of a strong lethal genetic disease, we might want to artificially fertilize those embryos and then look at the ones that don't carry the disease alleles and only implant those. About 20% successful if you're not yet 35, but it is tremendously expensive, $10,000 to $15,000. Gift or gamete intrafallopian transfer is when a woman takes a super ovulation drug. So she'll release a lot more than one oocyte. The doctor is going to take the largest eggs, mix those with the man's sperm, and then deposit them back into the fallopian tube. We're looking at a higher success rate than IVF because we're not fertilizing outside of the human body. We put the eggs and the sperm and just mix them in the body, but it is just as, if not a little more expensive. And then PGD. I sort of mentioned this when I was talking about IVF. After IVF, but before implantation, we can remove a cell from the clump of cells and test it for its genetic makeup. We can then choose the embryos that do not carry certain markers or genetic diseases and implant only those into the woman's uterus. There are some ethics issues here because potentially you could select for traits like biological sex or physical characteristics. But I do believe right now it's only legal for disease purposes. This can also be used to provide stem cells for a sibling suffering with a similar inherited disease, or even banking these stem cells in case the eventual individual or baby has issues in the future. Success rate, 99%, cost $5,000 to $10,000.